Alrighty, here we have the electron transport chain. We have the inner membrane space designated on the yellow side and the mitochondrial matrix designated on the pink side. Separating the two, we have the inner mitochondrial membrane, which contains many proteins. The first of which we're going to discuss is NADH dehydrogenase, which oxidizes NADH. This protein is shown in blue, located in the beginning, labeled Roman numeral 1. Moving forward, we have protein 2, labeled in green, which is a peripheral protein known as succinate dehydrogenase. This is where we find FADH2. Moving forward, we have protein 3, which is an integral protein, and it's known as cytochrome C reductase. You'll see this on this diagram colored in purple, labeled Roman numeral 3. Moving forward, we have the fourth protein located to the right, and that's colored in red and labeled Roman numeral 4. This protein is known as cytochrome C oxidase. Cytochrome C, shown in blue, is a peripheral protein shuttle that carries energy and electrons from proteins 3 to 4. Ubiquinone, shown as a pink Q, is a shuttle of energy and electrons from proteins 1 to 2 and 3. It's important to note that proteins 1, 3, and 4 are protein pumps. This means as electrons are pulled through, protons are pumped into the intermembrane space. ATP synthase at the end uses the gradient, which we'll talk about later. Talking about NADH and FADH2, they're both high energy electron carriers. They act as shuttles, or as some like to refer to them, taxis, but they differ in where they drop electrons off. They both must be regenerated, which means simply that they must go get more electrons and continue carrying them. But NADH carries hydride ions, which is 1 proton and 2 electrons, plus that additional proton. The 2 electrons go to the pump, and that causes 4 hydrogen ions, or protons, to go into the inner membrane space, as shown. Moving forward, we have FADH, which drops its electrons off at succinate dehydrogenase, which is not a pump. So it doesn't make as much ATP as NADH, but when it drops off its electrons, they're shuttled by ubiquinone, the pink Q, which takes electrons to the third complex, which then allows the 4 hydrogens to be pumped. Moving forward, keeping track of where we are now, we have a grand total of 8 NADH protons and about 4 protons from FADH2. So NADH is still in the lead. Electrons then go to cytochrome C, where they're passed to protein 4. Keeping up a count now, that would lead 12 protons pumped by NADH and 8 protons pumped by FADH2. The 2 electrons meet with some protons, providing at cytochrome 4, you have 2 hydrogen ions and half of an O2, producing water. This water is known as metabolic water. And we put this half here in front of the O2 because O2 is found in an environmental state, but we only need one of the oxygens for water, so that's where the prefix of half comes from. Moving forward, we are going to talk about how the electrons come down with the concentration gradient and the electrochemical gradient, like their behaviors tell them they want to. The only spot for them to do this is ATP synthase. A way to think about this is ATP synthase drives this molecular motor, and this is where you find ADP combining with an inorganic phosphate, and this is where we're getting that output of ATP, which we know is an energy molecule. What's important to remember is every NADH, there's 12 protons pumped, as we discussed previously, which equals about 2.5 ADP. For FATH2, we only have about 8 protons, which only yields about 1.5 ATP. It's important to keep in mind that for fatty acids, there's much more FADH. Altogether, this is the electron transport chain, part of cellular respiration, as diagrammed.